Thank you, everybody, for joining us for our first invited talk session. I have had the opportunity of knowing Nan Laird since my time years ago at School of Public Health at Harvard in Boston. And she has become extremely well known for her incredibly important and impactful work on clinical trials and meta-analysis of clinical trials, which really has influenced the way that we look at the safety and efficacy of whatever intervention has been attempted in that clinical trial, whether it's a medical pharmacological intervention, whether it's a surgical intervention, doesn't matter what the treatment intervention may have been in the clinical trial. We have to be able to examine the statistical data analysis results in an unbiased manner that does not introduce misinterpretations. Professor Nan Laird at Harvard has had a huge impact. So it's with great pleasure and great thanks and appreciation that I welcome Professor Nan Laird to address how to analyze results from genome-wide association studies. And to help her out this morning, we're going to start first with a presentation by Julian Hecker, who is now also at the School of Public Health at Harvard. And he's going to lay the foundation for how we look at these GWAS before then Professor Laird gives her presentation. So let's start with an introduction to GWAS and how we examine the results. Dr. Hecker?
Yeah, I'm starting to share my screen. Can you see my screen now?
Yes, thank you.
Okay, let me go to one second. Can you see it now still?
Yes.
Okay, perfect. Yeah, so thank you so much for the introduction. I'm Julian Hecker. I'm an instructor in medicine at Harvard Medical School and an investigator at the training division of network medicine at the Brigham and Women's Hospital in Boston. And I was in the fortunate situation to work on some joint projects with Nan during my PhD and my postdoc. And before Nan's presentation, I would like to give you some introduction to genome-wide association studies called GWAS and their benefits and limitations. So I will mention a couple of methods and papers, and this is, please do not to be understood as a comprehensive list. It's really just some examples to illustrate the challenges and some proposed approaches in the field. So as a warm-up or introduction, so genome-wide association studies, or GWAS, are based on a concept that is actually pretty straightforward. So we test each single nucleotide variant or polymorphism, SNPs or SNVs called, for association with a complex trait or disease. And the number of SNPs tested and the density really depends on the underlying technical platform, and the field started to use chip arrays, then we used imputation, whole exome sequencing, and nowadays whole genome sequencing. So basically we have the information across the whole genome available, and this leads to more than 1 million tests performed in a GWAS nowadays, based on all common SNPs. So basically all SNPs where we have some substantial variation in the population. And because we perform so many tests, there's a multiple testing burden, and an established significance level is at 5 times 10 to the minus 8, which corresponds to a significance level of 5%, and adjusting for approximately 1 million independent tests. There's a lot of correlation in the genome. That's why this significance level is still quite often used and established because the number of independent tests is not equal to the number of SNPs tested. So the first GWAS was published in 2005, and there were a lot of skepticism about the approach, but so from my computational point of view, I would say that nowadays it's more considered as a success story. And the skepticism usually is around the observation that was made in the beginning, that most complex diseases are described by polygenic architecture. That means that in comparison to Mendelian diseases, there's not only one gene involved, but there are a huge number of genes and genetic loci involved. The effect sizes are small, and therefore we need large sample sizes to identify genetic associations at this stringent significance level. And in order to increase sample sizes, meta-analysis play a crucial role, and this demonstrates the very important contribution that Nan made to this field of genetic epidemiology, because meta-analysis is really a key concept in modern GWAS. As I said, because there's this polygenic architecture, there's also the observation that there were previous studies, usually based on twins, that inferred the heritability of some traits. The heritability is defined by the proportion of phenotypic variants that can be explained by genetics. So basically, we have some number of the fraction that we can hope to explain by GWAS findings, but the actual number of or the actual fraction of variants that could be explained by the identified GWAS association so far was usually much smaller. And this led to this gap between, or hidden heritability or missing heritability between GWAS findings and the twin study heritability estimates. But since the sample size increase and we identify more and more associations, this gap is getting smaller, or is to some degree even closed for a trait such as human genetics, human height, where a lot of independent genetic loci were established. And of course, overall, the goal of GWAS is to identify disease mechanisms, to identify drug targets, and to improve personalized medicine by incorporating the genetic profile of a patient in order to understand the individual risk, or maybe the personalized treatment that could help this patient more compared to a general approach. And if you look at GWAS publications, you will often see, or almost always see, a Manhattan plot. This is an example of a Manhattan plot from a recent publication, and it's called Manhattan plot because this basically resembles the skyline of Manhattan. But I have to say, given the sample sizes nowadays, these peaks are getting higher and higher. And the peaks here are the negative logarithm of p-values, and you can see that these basically visualize all the associations across the genome, partitioned by the 22 chromosomes usually, and this vertical line corresponds to the significance level. So basically all peaks above this line are considered as genome-wide significant hits in the GWAS. And if you zoom in to a small genetic region, this is from a different publication, so it does not correspond to the previous Manhattan plot, but it's just as an illustration. So if you zoom in, you can look at a regional association plot where each dot corresponds to a SNP tested. And the black dot here corresponds to the index SNP, so the SNP with the smallest p-value in the GWAS in this loci. And then the other points are colored by their LD measure between this index SNP and the other corresponding SNPs. And if they are red, this means that they are in high LD. LD is linkage disequilibrium and is defined by the non-random association between nearby alleles due to a linkage. And this illustrates you that if there's a genetic hit in a small genetic region, we usually do not observe only one association, but a signal cluster. And we will talk about this and why this is a problem in a minute in more detail. And this figure here is also a nice illustration from a recent publication or review article, and it illustrates the relationship between sample sizes and the number of independent genetic loci established. So we are not counting the LD associations here, but the independent loci across the genome. As you can see, for BMI and height, sample size really matters and increases the number of identified loci. But as the title suggests, I would like to talk about some challenges and limitations of the GWAS approach, and I partitioned this into three sections. The first is confounding, or as you could maybe want to put it, correlation is not causation. So there are two issues. So population stratification is a phenomena that I will explain in a minute in more detail, and this can introduce false positives to a GWAS. And the second is that, as you saw on the region plot, if we identify a genetic association in a genetic loci, there's not only one finding, but a signal cluster. And if we are interested in identifying the causal variant, we have to take into account this LD structure in order to understand which one is really the causal one. Because many of these variants, because they are all tested separately, will show genetic association, but this does not imply that they have a causal effect on the phenotype. The second section is technical because all GWAS nowadays are performed using software solutions that are usually implemented by research groups. And of course, implementation of a methodology is very helpful to push the impact of a publication, but there's usually not much resources to maintain these software solutions. And because this is actually nowadays very challenging because the sample sizes go up to, let's say, 1 million samples, these software solutions are getting very complicated and sometimes bugs or errors can occur. This, of course, is also a technical problem in GWAS. And the third point, I'm calling this here GWAS to biology, is, of course, given that we can exclude the first two sections so that there's no confounding, there's no technical error, but we have an established significant association in GWAS, we want to understand what does it actually mean for the disease. Can we identify disease mechanisms? And as I said, identifying the causal variants in a genetic signal cluster is challenging, but identifying those corresponding genes or mechanisms is even harder. So the first point, confounding. So what is population stratification? We know from population genetics that due to genetic drift, there are allele frequency differences between populations. And if we have a case control study, that's one of the typical study designs, it might be the case that actually cases and controls are sampled with different fraction from these two populations. So it's an example here with two populations. And if you sample cases and controls with these different fractions, this could lead to different genotype distributions for a specific genetic variant when you compare cases versus controls. And if you test for an association between the genotype distribution and case control status, you would get a significant finding maybe, even though this genetic variant does not have a causal effect on the phenotype. So it's a false positive, just due to the fact that because there's allele frequencies between these two populations, and cases and controls were sampled with different fractions from these two populations. And a common approach that is used in the field is based on principal component analysis. So we take the whole genetic information across the whole genome, perform a principal component analysis, and identify the systematic pattern across the whole genome, and incorporate these principal components as covariates in the GWAS regression. And this actually works pretty well in practice for most cases and can adjust for this population stratification. And in these plots, you can see if you perform a principal component analysis based on genetic data, you can really identify these clusters of genetic ancestries. And that's the key to adjust for these population differences. But there's also another approach, and it's based on the design of family-based association studies. And there was this approach called TDT, Transmission Disequilibrium Test, in 1993. It's based on trios. So you have genotype data for the parents and an offspring, usually an affected offspring, so a case, if you want to say it like this. And then, given the parental genotypes, we can model the offspring genotype by Mendelian laws. And because these Mendelian laws and the probabilities do not depend on any underlying frequency assumptions or any distribution assumptions about the parental genotypes, this type of approach is completely robust against population stratification. This was a very important approach that was extended by Nan and others to the family-based association test framework, FBAT. And she was able to extend this to general pedigrees, other phenotype distributions, haplotype approaches, and this is based on a very elegant mathematical approach called sufficient statistics. And this illustrates actually the second out of several very important contributions that Nan had that made to the field. The second part of confounding is due to ID. As I said, if you look at this regional association plot, we have a couple of genetic associations that are usually all significant, and we would like to find out which one is the most likely causal variant. And this is actually a very challenging task, and there were a couple of methodologies proposed to perform this type of fine mapping. So basically going from this signal cluster to a small number of candidate causal variants. And these approaches are usually based on purely statistical models that take into account the intrinsic LD structure at the loci. But as you can observe, this resolution level is lower bounded by this LD structure because we cannot distinguish between two associations based on two variants that are in basically perfect LD with each other. So then there's no way based on a purely statistical approach to distinguish which one is the causal variant. And the second part of technical challenges or challenges of GWAS is technical errors. And here I have an example that is actually related to one of my PhD projects. And I want to make this clear. This is not about blaming anyone. It's really just give an example that these things can happen. There is a gene-based approach for genetic association testing called VEGAS or VEGAS-2. And it tries to combine this single variant test into a gene-based test by incorporating all variants that are mapped to this gene, and then trying to test these SNPs jointly for association with the phenotype. So it's basically an approach that tries to gene map these associations. And this was published in 2 papers, and during my PhD, I was actually discussing with other PhD students about this approach and how this is implemented in the software. And then we found out that there is a small mistake in the code. And it's really just a single bracket that was misplaced. And this leads to a deviation from what the tool does compared to what is actually described in the paper. And it's a very tricky error because the output of the tool was actually still reasonable. So there was no way to see, okay, it's completely off, there must be something wrong. It was a very subtle error, but we were able to show that this can introduce false positives. And we reported a correction for this, and this was also incorporated in this tool. And again, to say, this is something that can always happen if someone has any experience with software development. This is something that always happens, and it's just very hard to maintain and test these software solutions, and especially when there's no additional funding or any resources to do this, because there's of course always an ongoing pressure to work on new publications and stuff like this. The last part, and of course I have to mention here again, so I'm coming from a computational mathematical background, so this is my perspective here to some degree. When we talk from the step from going from GWAS to biology, one of the major observations in the field, and I think that's also why there's some skepticism about this approach, is that majority of GWAS associations so far, and there are like hundreds of thousands of associations reported, most of them remain mechanistically uncharacterized. So we do not understand the exact underlying mechanism of these associations. And this also made more complicated by the observation that most GWAS hits are located in non-coding regions of the genome, and it's the working hypothesis that they are involved in gene regulation effects, but it's very hard to infer the corresponding genes and the exact tissues and cell types, and this is an ongoing challenge where there's a lot of emphasis on right now. So the first approach to do this is using function annotations. There were a couple of projects in the field that try to establish function annotations for variants, and this was actually pretty successful for severe class gene disrupting variants. But for most variant classes across the genome, these predictions are actually very hard to make, and that's still an ongoing, I would say, like there's still a lot of work ongoing on trying to improve these predictions. The second approach is using quantitative trait loci studies, QTL. And in this QTL approaches, we try to identify genetic associations between variants and molecular traits such as gene expression, splicing, or chromatin accessibility. The most famous example is surely gene expression levels, so it's called cis-eQTL studies, where we try to identify this association between nearby variants and gene expression levels. And this GTEx consortium is I think one of the most popular projects that was published and reports many cis-eQTL associations in different tissues. But again, in general, this approach is limited by the available tissues. So most of the studies are based on blood because it's actually pretty accessible. But if you're interested in disease mechanisms, usually the cell types and tissues are very important, and also the timing sometimes when these samples were drawn is important, and this is, compared to the universal character of the DNA, very challenging to identify the right QTL mechanisms at the right time, the right cell type. New directions here include single cell sequencing and spatial transcriptomics. And there are also some purely computational based approaches. I picked here two recent papers that try to learn SNP to gene mappings from established and reported GWAS. And they try to incorporate different functional annotations and eQTL results, and also some like very basic annotations like enhancer and transcription factor mappings to learn SNP to gene maps. And this is also an approach that tries to improve the links between these genetic associations reported in GWAS and actual biological mechanisms. So next steps and outlook, I would say biobanks are getting very, very important because they have large sample sizes and a lot of collected phenotypes. The most popular or famous example here is the UK Biobank, which like 500,000 samples or participants with a lot of deep phenotyping available and genomic data. Then historically, the initial GWAS were usually based on European ancestry samples. And there's a huge push at the moment to incorporate also non-European ancestries in these GWAS. So to increase the diversity in these studies. And I think that's also one link maybe to Nan's presentation, because I think she wants she will also talk about the fact that heterogeneity is helpful. For example, if you think about this fine mapping problem, that because there are allele frequencies between populations, there are also differences between populations. And if you incorporate different genetic ancestries, this might help you to infer the causal variance, because you have different ID structures and maybe can improve this resolution level of these fine mapping approaches. Then one approach is also to incorporate multi-omic data, for example, in the TopMed initiative, where a lot of different omic layers are collected, for example, gene expression, metabolites, methylation, and proteomics. And in order to link the genetic association with the phenotype through these individual layers and to identify additional biomarkers for disease. And also mentioned single cell and spatial transcriptomics are getting very popular at the moment and very important to the field. And as I said in the beginning, or I just mentioned, I'm more like coming from a computation perspective. So I did not cover here approaches that are of course very important, like cell models or animal models or gene perturbation approaches. So where we really try to, or researchers really try to identify the functional mechanisms in the lab or in experiments. But of course, for complex genetics, this is also quite challenging because, as I said, these effect sizes are quite small. So it's definitely something that needs to do. And of course, the final step to really understand the genetic association is if we can identify these mechanisms in cell or animal models. And one thing, again, to emphasize why GWAS I think is important is this citation here, or this quote that I put in this box. It's from a paper that is actually talking about challenges in psychiatric genetics, but I think it also applies to other diseases, is that drug targets with human genetic evidence are twice as likely to lead to approved drugs. And this can be even more increase the chance by knowing the exact causal genes that are involved. So thank you so much. I hope this was some kind of introduction that is helpful to lay down the foundation for Nan's talk. And now I think I and we all are very excited to looking forward to Nan's presentation. Thank you.
Thank you so much, Julian. Fabulous presentation. Very much appreciate your help here and your contributions in setting up the background so that we can now hear from Nan. I do have your slides on my screen, and I hope they're visible to everybody. Please.
Yes, you can see them.
Excellent, thank you. So Nan, please.
Okay, well, I want to start by thanking Julian very much for excellent presentation that saves me a lot of trouble in explaining these concepts. And also thanks to Carl for inviting me. Next slide, please. So I'm going to give just a little bit of background on meta-analysis. As Carl said, I first started, got interested in meta-analysis back in working with clinical trials, actually even before then. And there is a formal definition, and it arises in the context where you're studying a phenomenon, like does this gene influence obesity? And there are many, many studies out there of whether or not this gene influences obesity, and you'd like to just summarize the results. And a formal definition called meta-analysis was given by GWAS in 1976. And it's basically, it's the statistical analysis of the findings of a collection of individual studies. So it's different from simply doing a review and saying there were 20 studies and five of them were positive and 15 weren't. It actually uses a quantitative summary from each study and uses that to get an overall result. Next slide, please. So just a little background, the meta-analysis was used very extensively in the 1970s, and people were really in the social sciences, and people were interested in really big questions like, is psychotherapy effective? And they didn't want to talk about, well, this kind of psychotherapy or that kind, or for this kind of illness or that kind. They just want to know, well, can you say it's been effective or not? Or what is the optimal class size for learning? There were many studies of this in elementary schools. And another one was, are there teacher expectancy effects and how big are those effects? People were quite interested in having a quantitative study. And one that I think we are all probably familiar with today that comes from clinical trials was the study by Beecher in 1955 of the placebo effect. Next slide, please. So this is a result which I can barely see myself, so I'm going to presume that you don't have to look at the details of this, but he collected quite a few studies of all different kinds. There was many of postoperative wound pain, and there were some of cough or drug-induced mood changes or pain from angina. And in each case, there was a treated group and a placebo group. And so all of the untreated groups did have a placebo, and he lists the different conditions as well as the different placebo agents, and looked at the number of patients in each placebo group and the number that were satisfactorily relieved by the placebo. And actually, there's a range, but there's a substantial, surprisingly large people that are relieved by a placebo and across these different conditions and across these different placebo agents. And the average level is about 35%. And that's sort of taken as typical for the placebo effect across many different medical disciplines and treatments. Next slide, please. So that's one of the earliest meta-analyses. But the early methods were, apart from Beecher's, were largely descriptive, and people would report on the studies and what were the characteristics of the patients and what were the characteristics of the treatments. But the meta-analysis is different in the sense that you want to get a summary from each study. And much of the early work was, how do you describe that study, that summary that you want to take, and then how do you combine those effects to give the overall summary. And there was much discussion, but the discussion was what to do about heterogeneity. How do you identify it? And what should you do? Many people taking the point of view that if you see evidence of heterogeneity in the studies, you shouldn't combine them. Next slide, please. And this is one of my favorite slides. I'll give you just a minute. This is an important slide to read. And you can see why people would be against combining quantitative measures if you're talking about very quantitatively different measures. But in the context of a meta-analysis, we are thinking about taking a quantitative measure from each study, which has the same basic interpretation. So next slide, please. So I'm just going to introduce a little bit of notation, not much statistics here. So I'm going to assume that there's an effect size for each study. And in the context of many of the genetic studies, it's going to be a risk difference between treated and control, or between an odds ratio if you have a case and control study, or with a quantitative trait, it might be a mean difference, or it might be a regression coefficient. And we are also going to have a standard error denoted by S squared of Y. So it's a measure of variability of that, the variability in that study, the variability of the Y. Next slide, please. And there was a method and a model introduced by Cochrane in 1954, which I call a random effects model. And he actually introduced this for the results of field trials. So it's a very old model that was used successfully for combining results of different agricultural studies. But the reason it's called a random effects model is that you assume that the effect of the true treatment is TI might be different for each study, and EI is the random variation around the ith study. And so the sampling variability for that is what I called earlier S squared or SI squared for the ith study. But you're interested in measuring an overall effect, that is the mean of all of the treatment effects. And we could quantify the extent to which the studies vary from each other by variance sigma squared. So this sigma squared is different from the S squared. Sigma is telling you how much the studies are varying from each other. And the S squared simply tells you about how much noise there is in the ith study. And that often depends quite heavily on how big the sample size is in the ith study, but it can also depend upon the design of the study. So next slide, please. And the method for analysis that was proposed by myself and Rebecca Dersimonian in the context of clinical trials was to just use a weighted estimator. So I'm not going to go through everything in this slide, but basically we proposed a method for estimating the overall mean, and it's just a weighted mean where the weights depend not only on the within study error, but it depends upon estimate of the sigma squared, which is the between study variation. And we proposed a method for estimating the variance, and we also proposed a method for estimating sigma squared. So the next slide, please. So the random effects approach to meta-analysis, which I'm going to talk about today, has the advantages. First of all, it provides a quantitative measure of how the results differ from study to study, which I feel is always an important thing to do when you're doing meta-analysis, is assess the variability in the results. And this is variability above and beyond the sampling error that's present within each study. But some criticisms, as I've alluded to before, is that some people feel that there shouldn't be any overall combination of the results if the effects do differ. But I personally have a different perspective. My perspective is that not only do you want to provide an overall study, but you want to characterize the variability that's present and report on the variation in the study results. And I should add also that often goes hand in hand with elucidating the effects of some major differences that you can see between the studies in terms of, for example, study design or perhaps study population. So next slide, please. The fixed effects model, this is in contrast into the random, is essentially you're saying, no, all the studies are measuring the same effect. So it's the same as you set your sigma or your sigma squared equal to 0. And then you're going to estimate your mean with a weighted sample mean. But now the weights depend only on the variation within the sample. So it's a weight driven by the sample size in each study. And the estimates turn out to be pretty similar. I'll give you some examples of that. But the confidence intervals for the overall effect are usually larger, and in some cases substantially larger if you take a random effects approach. Next slide, please. So I'm going to give you an example of meta-analysis in a GWAS study. Now, in a typical GWAS study, but certainly not always, typically it's a case control study which compares genetic variants in the cases and the controls. And Julian has talked about many of the issues involved in a GWAS study where you might have millions of genetic variants being tested, and each is tested separately. And the primary interest in the GWAS is testing for genetic effects, not the size of the effect. But the effect sizes can be extremely small, and the typical significance level also extremely small. But there's a lot of interest in meta-analysis because for increased power and replication. But many individual studies are small, thousands of individuals, but the numbers of studies may be small. That's another feature of genetic meta-analysis. Next slide, please. So I'm going to talk about obesity and a variant, and it's not a variant in the gene in situ. It was a variant close to in situ, a SNP, which was found in one of the very first GWAS in the Framingham study population. It was found to be significant. So in the original paper, because it was somewhat controversial and using the kind of controversial approach to a GWAS, it was replicated in the original paper. We had 6 different cohorts besides the Framingham study, and that SNP was tested in all 6 of them, and it was replicated in 5, but not 1. But we did some follow-up on this, and it was of particular interest to the original investigators was the design of the study. Because in the samples that we had, there were 3 different types of studies that were done. One was a general population study. So the Framingham heart study is an example of a general population study. As many of you may know, it was started back in the 1950s, and it was done from a random sample of households. People were not excluded on the basis of disease of any kind or age or health status. I guess it's not random in the sense it didn't include people in institutions, but otherwise a pretty random sample of the Framingham population. Another type of population where we have family cohorts that are, general cohorts of the population that are available was nurses' health study and the physician health study. So these are cohorts of individuals such as working employed nurses, employed physicians who by virtue of being employed were healthier than the general population. Or then a third type of study that's commonly available for testing is a case control study where people had drawn cases of people who were obese and then controls from the same population, but who were not obese. So the next slide, please. This is actually the original study. So I won't go through this, but it just shows you the Framingham heart study, FHS, which was the original study, was a family design. And then the rest were different studies that were recruited to test this one SNP that was found significant in the original study. The next slide, please. But after this study came out, there was a lot of controversy about this paper for all the reasons that Julian has mentioned earlier. A lot of skepticism about association studies, well, rightfully so, I would say. But there was controversy about this paper. And a feature about studying obesity is everybody has a measure of obesity. You can get body mass index, or you can get whether or not the person is obese. So everybody jumped on board, and a lot of people actually wrote letters to the journal saying, well, I have a sample of cases and controls, or I have a cohort of nurses, and I compared the obese to the non-obese using this SNP, and you only had to genotype one SNP. So we had many published studies, many of which were not true studies. They were just letter to the editor. And so we undertook a meta-analysis of these studies. We collected the results from 34 publications that did not include the original 6 publications because we thought that would bias the results. So we took 34 publications, and they included 74,000 subjects. To look, our primary interest here is to ask, well, what about the effective study population? So the next slide, please, Carl. So again, we looked at 3, the general public's population study, or sometimes called a cohort, and the healthy population. Again, it's a cohort, but a cohort that's been selected, sometimes selected for disease, but usually selected against disease, or then subjects selected basically on the basis of obesity or not. Next slide, please. And these are a pictorial summary of the results of giving the odds ratios for each of the studies by category. Down at the bottom, let's look down at the bottom. The overall test for heterogeneity right down at the bottom was a chi-square on 13 degrees of freedom. I'm sorry, 5 degrees of freedom with a P of 0.02. And I-squared, that's the percentage of the variation that's not explained by the within study variation. So that indicates overall the results of the study are quite heterogeneous. And if you look, however, within each study, there's the first is the general population adult studies. And by the way, I should say all of these were Caucasian subjects, just to eliminate one particular type of potential differential. And you can see that there is variation here, but actually almost all of the odds ratios confidence intervals include the vertical line, which indicates that except for the last 2, which were large general population studies, very large, they were positive, but the others, either they tended to be positive but were not significant, or a few that were negative, but again, were not significant. So overall, the indicator, this I-squared is an indicator of the percent of the overall variability that's explained by the within study variation. So it's rather small in this case, so it's an indicator of not much heterogeneity here. And the pooled odds ratio is positive, but relatively small. When you look at the healthy populations, it's interesting. They're all negative, and the overall combination is got a large confidence interval, but it is overall negative. But it has no heterogeneity here. They're all negative. And then when you look at the obesity analysis, overall, they're mostly positive, and although there's a fairly large confidence interval, so those pooled results are not significant. Now, why would you expect this? For one thing, I think, let's look at the next slide. I think maybe I have some comments on why on the next slide. Oh, this is just another summary of the results. This summary of this results shows the difference between your estimated fixed effect and your estimated random effect. And as I said to you, they both estimates just use weights. There's not much difference in the weights. So the estimates themselves are pretty similar. That is looking at the odds ratios in columns 2 and 3. But if you look at the I-squared, that's the index of heterogeneity. But also I want to draw attention to the p-value here. You see that in the second and third columns, if you look at the p-values, you see that the effect of the random effects is to include more variation or account for more variation, so that the overall p-values are tend to be less significant, although that's not actually true for the healthy population where they're the same. And that's because there's no heterogeneity evidence in this sample. But in general, the random effects is more conservative. So let's look at the next slide. And before I talk about the next slide, let me just comment about why it's not surprising that the healthy population might not replicate a finding from a GWAS. And that's because these people have been excluded from consideration. So you won't find people with a variety of different types of common illnesses or chronic illnesses in the healthy populations. So you would not necessarily expect them to be able to replicate many genetic studies. But in summary, I'd like to say that heterogeneity does not preclude combining results, but I think it is important to report a measure of heterogeneity. The I-squared is normally what people use. And if you're going to combine studies when there is heterogeneity present, I think it's useful to see if there are known factors that could explain the heterogeneity, and you need to have some caution in interpreting the results. But heterogeneity can suggest important insights as to the nature of interventions, populations, and study designs. Thank you.
Thank you so much, Nan. Thank you so much, Julian. I do think that we should try and take a few minutes for any questions. If you have a question, please make sure you turn on your microphone and your video. I have a question if there are none from the audience. Well, I'll go ahead then. Certainly, I'd like to comment just briefly on practical approaches to statistical evaluations from the very beginning of my early work in statistics, where I was mentored by Joe Berkson and Bill Taylor at Mayo Clinic. There was an emphasis on practical, operational, quote unquote, non-theoretical approaches to statistical data analyses. So I've always been a huge fan of simple, straightforward, weighted means, method of moments. These are very practical, but they also lead to an extremely relevant practical question here, which is the choice of threshold. So when you make a choice about what you consider a significant p-value or not for a statistical association of a SNP, how do you go about choosing that threshold for what p-value you consider significant or not? In general statistics, there have been a number of papers over the years that have argued against considering, we got to move beyond a p-value of 0.05. Let's go to a p-value of 0.001. let's go to a p-value of, order of magnitude, 10 to the minus 5. So let me put this question to Julian. How do we go about choosing the threshold for p-values on whether or not a SNP has a significant association or not?
Yeah, I would say that, of course, it's pretty established to talk about the overall significance level of 5%. But I think also what maybe building a bridge to what Nan said, I think nowadays it's all about replication. So it's if you try to publish a GWAS nowadays without any replication, it's very hard. So you need to make sure that you can always replicate these results in other studies. And I think that's why, of course, there's a lot of criticism of p-values themselves. So I think the goal would be to have enough results to not have a discussion if it's just a matter of how you choose a significance level or yeah, any threshold, I guess. Yeah. That would be the goal. I mean, of course, I agree that if, let's say, you perform a GWAS and the only result is one SNP with a p-value of 4 times 10 to the minus 8, which is just below 5 times 10 to the minus 8, yeah, it's not the strongest finding, I guess. And that's why you always try to include replication of results.
Well, I'm very pleased to hear that answer to the question, because it certainly supports the theme of our workshop today, which is, of course, reproducibility, reliability, replicability, integrity in our research. So if there's any other quick question, but if not, let me close this session, and we'll take a brief few minutes break so that I can stop this recording. I will certainly leave my camera on, and we'll restart the next talk in just a few minutes.